All right, now I'm back. It looks like everything's working, so we should have a actual lecture for you to look at. And now here's another one. The different membranes, like we showed you with the sphingolipids in the last thing, the different membranes can have different compositions. You can have a bunch of sphingolipids on the inside or the outside. They are distinct domains. They have distinct qualities. And so you can actually take, if you look at this, you can take the plasma membrane of a cell, and you can find out that it's got a lot of cholesterol in it, a lot of orange. It's got a lot of sphingolipids in it, a lot of green. The other kinds less. If you look at the inner mitochondrial membrane, you can see that there's actually a lot of red in the inner mitochondrial right here. That turns out to be cardiolipin, which I'll tell you about why that's in the inner mitochondrial membrane and not really many other places, because it serves a particular function there. So you can have different kinds of membranes in different compositions in different membranes, and you can quantify that and you can try to figure out why. You can also have differences in the inner and outer monolayers. On the last slide you had differences between the inner mitochondrial membrane and the outer mitochondrial membrane. Well, this shows you can have different sides of the monolayer, can even have different compositions. For example, if you look up and down, the dark blue bars show what's in the inner monolayer and the light blue bars show what's in the outer monolayer. You can see that sphingomyelin and phosphatidylcholine, those both point out. Other things like phosphatidylserine and phosphatidylinositol point to the inside. And so you can try to understand, well, why do these point to the inside? They actually sometimes are sending signals to the inside of the cell. The ones pointing outside are sending signals to the outside of the cell, or providing attachment points or something else that the outside of the cell needs to do. So the inside signals are the ones that are sort of on the bottom of this, and the outside signals are sort of on the top, with the exception of that very top bar, which is an inside signal. So membranes will flow differently depending on their lipid composition, and they'll curve differently. If you add cholesterol, they'll be able to curve more, things like that, and they'll be more or less solid depending on the amount of saturation you have. And so it's a really complex and interesting area to look at. And when you have a eukaryotic cell, you have a lot of different kinds of membranes that might have different kinds of properties. You can think about these properties in terms of chemistry if you can identify all the components and you can put them all together and you can say to yourself, okay, over here, this shows that the ER has a lot of unsaturated fatty acids in it, and it's going to have like not that much cholesterol, there's not that much of the blue parts, and it's about as thick, the carbon chains are long enough to make it so that a transmembrane domain that goes directly across it in an alpha helix sort of structure has 20 amino acids in it. And so that makes a certain kind of membrane. If you look at the plasma membrane on the bottom side here, you can see that there's actually a lot of charged lipids on the surface, on the outside surface of it. You have a lot more saturated fatty acids, and so that means it's going to be a lot more solid. It's going to have a different melting temperature. And also you have more carbons, and so it's going to be longer, it's going to be thicker, and it's going to take more amino acids to make a transmembrane domain that bridges both sides of the membrane, that sits through the thickness of that membrane. So these minor differences can have major biological differences in how the different membranes work. Turns out the ER membrane is good at doing what it needs it to do. So it needs a more fluid membrane for whatever reason. The plasma membrane needs to be sturdier, less fluid. The thing that we showed you on the 2 slides ago is that they have a distinct outside and inside. And the reason for that is because it's really hard for a lipid to go from the outside of a bilayer to the inside of a bilayer. Even though their feet are touching in the middle, the head group is difficult to get across. So you can look at that as 3 reactions. If you have the trans bilayer, or what they call flip-flop diffusion, if you have a lipid trying to move from the outer leaflet to the inner leaflet, the outer part of the bilayer, then that's going to be very slow because the transition state for that is really high. You have to be submerging the positively or negatively charged head group in a very hydrophobic environment. So hydrophobic environment takes a lot of energy. That kind of energy doesn't come along very often, and it's a very slow, it takes days for a lipid to move from inside to outside. That means it's not just going to happen. On the other hand, lateral diffusion, shown in part B, where the lipid is just moving from side to side within the same leaflet, That's very fast. A lipid can move like a micrometer per second. That means a lipid can move like all the way around the cell if it really wants to. So moving side to side is real easy. Moving top to bottom, real hard. But whenever you have something that's hard and you have a high transition state, you can make an enzyme that stabilizes that high transition state and catalyzes the reaction. So you can make these enzymes like they show on the bottom that will actually take the head groups and it will actually catalyze the movement of the head group from one leaflet to the other. And those are called flipases and flopases. It takes ATP, it takes energy to move just one lipid. But if you think about it, this is kind of like a symporter or an antiporter. So if you have two lipids that you want to move, you can actually make an enzyme that will move one lipid in and one lipid out, and that will actually use the energy from moving one lipid in to move the other lipid out. And this can actually sort of scramble the lipids without needing ATP at all. It's kind of a cool little trick. So just know that there are flipases and flopases that require ATP, but there's a scramblase that doesn't, and the scramblase is like an antiporter. There's an important experiment that proved that lateral diffusion is really easy to do. It's called the fluid mosaic model, is the theory that this lateral motion is really easy to do as opposed to the flip-flop transmembrane kind of motion, which is hard to do. And so how they proved this was actually kind of cool. They had a cell and they labeled the cell with red fluorescent labels on the outside. So they turned the cell red. The labels were all on the different polar groups, the different head groups. So what you did is you actually shone an intense laser on a small spot and you trained your microscope on that spot. You saw that the laser would do what's called photobleaching. It would actually take those red molecules and it would turn them white. It would bleach them white because of its intense energy, would basically break down the red color. And so that's that. If everything stayed in place, you would train your microscope on that and the white spot would just stay there. But instead what they saw was that the white spot turned pink and then it turned red again because red molecules diffused into the white space and the bleached photomolecules diffused out. They could time that and they could figure out how fast a single membrane molecule would move. Turns out that it moves really fast. Turns out that if you do this, you end up with the cell like sort of scrambles the membrane lipids around pretty quickly, and they can move into the area, showing there's a lot of lateral diffusion, but it's only lateral. It's within that particular bilayer, and it keeps the outer bilayer on the outside, but they can move around because, again, there's only weak hydrophobic interactions holding those things together. Oh, and another way to think of this is if you have different temperatures that you're introducing, as you increase temperature, you're introducing disorder into the membrane, and you're making it more liquid. It's just like melting. You can melt the membrane as well as you can melt a piece of beef fat or whatever. So let's say that you have a low temperature in part A. You have all of the fatty acid chains, they're packed really well together. They're packed tightly together. They're not moving as fast. If you heat it up, you get part B, where you have the fatty acid chains moving around a lot more lateral diffusion, and more disorder structurally in the middle of that membrane. That means it's more liquid-like. And this is actually part of the reason, the liquidity of the membrane is part of the reason why trans fats are bad for you. Or at least, I'm going to put it more positively, I'm going to say membrane fluidity is why cis fats are good for you. Because cis fats, remember that cis fats are kicking out. Because they're kicking out, they are more liquid, and they will end up with a more fluid membrane. This comes from the observation that if you give rats olive oil, it will lower their blood pressure. So what you do is you feed rats cis-oleic acid, which is naturally in olive oil, trans-oleic acid, which is the same stuff but flipped around to be the trans version, or soybean oil, which has no oleic acid in it. And it turns out of those three, the cis-oleic acid rats have lower blood pressure. And they looked for why. And the reason why is that the cis-oleic acid made the membrane more liquid. Because again, is kicking out like that, right? So that means it kicks out, it kicks the other molecules around, makes them pack less well. So at the same temperature, the same membrane is going to be more liquid if it has more cis as opposed to trans. The trans pack together better and they're more solid. They hypothesized that the structural reason for this is that the cells are more receptive to signals that are sent to reduce blood pressure. And they found out that this, there's actually some biophysical experiments to back this up. The more liquid membranes allows the cells to be more receptive to signals that are lowering blood pressure. Therefore, cis fats lower blood pressure. It's also part of the reason why olive oil might be healthier for you than beef fat, because it has more of these cis unsaturated fats in it. You can have an experiment like this where you take E. coli and you grow it up at different temperatures. It will make different membranes to survive well at that temperature. And as you increase the temperature, the E. coli that is grown up at the warmer temperature will have more unsaturated, actually I said that backwards, so that's why I should edit myself here. The E. coli that is grown up at the colder temperature will need to have a more fluid membrane at that temperature. It's going to be harder for it to have a fluid membrane, and so it's going to introduce more cis unsaturated amino acids. If you look up and down this right here, you have basically the proportion between 16, 16.1, and 18.1. And as you move from 10 degrees, you're going to see that you have more of the 16.1 and 18.1 at 10 degrees than you do at 40 degrees. At 40 degrees, you have a lot of the 16.0. That means colder E. coli make more membrane lipids that have more unsaturated double bonds kicking out in them to keep them liquid at the lower temperatures. At 40 degrees, they don't need to do that. They're liquid, they're kept liquid by the higher temperatures themselves, and so they can make more fully saturated fatty acids, and so they do. This ratio at the bottom is what you really want to look at. The ratio of unsaturated to saturated is about 3 to 1 at 10, and it's about the opposite, it's about 1 to 3 at 40. So look at this, and you have some homework based on this as well. If you think about this lateral diffusion in the complex case of the cell, where you have all these lipid rafts and membrane lipids and things like that, you can think about a case where if you do like the fluid mosaic model experiment, you can see that there's actually some areas of the cell where it looks like there's different regions, like the cells divided up into different states, kind of like the US is divided up into different states. For example, if you trace the movement of a single lipid, you'll start over here in the purple region, and you'll see it stays in the same purple area for a long time, and then it will jump over to the blue area. Then it will stay in the blue area for a little while, jump over to the green area, and then it will jump over to the yellow area last. Each of these areas is sort of a distinct area. So it looks like there's different components to the cell membrane. What could be causing this kind of behavior where you have kind of 4 different states, 4 different yards where the lipid can run around in, and there's some sort of fence between the yards where it can jump over when it goes from purple to blue or blue to green? Well, it looks like the lipids can move around to the different yards as long as it looks like there's different fences underneath the cell, not on the top of the cell. If you look at this, underneath the cell there's actually a network of proteins. Ankyrin and spectrin, for example, are 2 examples shown here, and actin is part of the complex that holds this together. There's this protein cytoskeleton that acts like our actual skeleton, but this is made of proteins underneath the cell, and the proteins form this sort of spider web arrangement. So you can see that you can have the lipid, and the lipid is sort of held in place by the cytoskeleton, but it can jump from one cytoskeleton place to another as it moves around. So this is a sign that there's just a lot of structural heterogeneity in the cell. You have some proteins that you look at will move around in the cell membrane. Some proteins will never move. Looks like those proteins are actually covalently bound to the cytoskeleton. So you see glycophorin and chloride bicarbonate exchange proteins in this example are actually covalently bound. They stay in one place. So the cell has a cytoskeleton to give itself some shape. It sort of molds the membrane, and there are protein movements that cause all these things to move around or to be tied down. And studying this is one of the cool things about cell biology. You can study what's tied down and what's not. The important thing is that if it's not tied down, it's basically fluid. If it is tied down, it's more solid. And so the fluid membranes, the thing about that is that you can have two membranes and you stick them together, they can dissolve into each other, just like two liquids can dissolve into each other. When you have a small little bubble inside a cell, you call it a vesicle. But it's easy to make a small little bilayer of a bubble. You can just make it inside a cell or outside a cell. When you push two of these membranes together, they can dissolve in each other, and so you can have it's called exocytosis if you have a vesicle inside a cell that moves to the outside of the cell. The reverse is called endocytosis, where you have sort of a bubble of a cell that's pinched off and it forms its own little vesicle with its own little bit from outside the cell. A lot of the movements that you're used to that are fluid and lifelike, they come from this membrane biochemistry, and they come from the hydrophobic effect and the fact that these bilayers are liquids essentially, and they move like liquids in life. Many of these processes are hugely important to how life works. And so you can have all of this works by membranes dissolving in each other. Another thing is if you punch a hole in a membrane, the membrane will seal that up by itself with a chemistry that's like this. It will flow together just like a liquid will flow together. It just happens in 2 dimensions. Membranes are really amazing biochemistry things. So if you look at a transmembrane protein, you can figure out a lot of things about it, even just from the primary structure itself. You can just read the amino acids. You might have seen some of these things in your bioinformatics project. So for example, if you have a 131 residue protein over here, you can look and you can see you have a lot of hydrophilic residues from 1 to 62, and then you have a lot of hydrophobic residues from 62 to 95, and then from 95 to the end you have a lot of hydrophilic residues again. From the pattern of all these hydrophobic residues, you can guess that the hydrophobic spanning part of the protein is from residues 60 to 95. You can guess a few other things about that. You can guess how wide the membrane is and how many residues would you need to go from one side of the membrane to another to form an alpha helix. You know the size of an alpha helix, and you know how many residues fit in that. The same rules apply here. So the other thing you can see is you can look for things like where are the sugars attached. Remember how we talked about how sugars are attached to particular sequences of amino acids and also outside only, not inside. Well, you can look for these kind of sequences. You can look for these codes in the protein sequence. And right here you can see that you have a bunch of these serines and threonines and even an asparagine here, all outside the protein. And so you can say that I think that my protein is outside from residues 1 to 60, is inside the membrane from 60 to 95, and is inside the cell from 95 to 131, all from looking at the amino acids. So notice how hydrophobic residues can embed themselves in a membrane. They can dissolve in a membrane just like hydrophilics can dissolve in water. So when you look at membranes, it's our last topic here. Some membrane proteins are sort of sitting on the membrane. We call those peripheral. And some membrane proteins are embedded in the membrane. We call those integral. The last protein on the last slide was an integral protein. Some proteins don't have a hydrophobic part to them. And these are 2 different types of proteins depending on how hard they are to remove from the membrane. If you see the integral membranes, the only way you're going to get those out is by adding detergent. You see how the detergent right here is solubilizing, forming a fake membrane and sort of allowing those hydrophobic residues to be covered up by the hydrophobic part of the detergent. On the other hand, peripheral proteins don't require detergent to remove. All they require is a little bit of salt or a little bit of pH change. This indicates that they are held there by ionic interactions, by some kind of charge interaction, and that means that they are just sitting on the head groups as opposed to being embedded in the membrane. So you see right here, the other thing that you can use is sometimes they're bridged by having calcium holding all the charges together. Well, calcium itself can be taken away by a chelating agent like EDTA. I think you'll have some example of this in your homework. But EDTA is a chemical that binds calcium. So if you add a chemical that changes the charges or takes away metal charges, does something to the charge, and if that causes the protein to leave the membrane, then you can be pretty sure that it is a peripheral protein. The other type of protein that you can have a protein that is actually attached to a lipid anchor. And the lipid anchor itself dissolves in the membrane. And this is called a GPI-linked protein because the glycophosphatidyl'inositol is the type of lipid that is actually in the membrane. Well, remember phospholipases? Phospholipases break down the lipid anchors. And so phospholipase C will be an example of an enzyme. If you apply that to the protein and you see the protein leave the membrane, then you can say, I've got a lipid-anchored protein. In fact, you can say, I know it's linked through this particular GPI linkage. It's a lipid. It's a special kind of lipid. We'll show you that in a second. Here's an example of a peripheral protein. It's interacting with the charged head groups. It's also interacting with purple calcium ions. You see these purple calcium ions here. And you see you have these phosphatidylcholine groups. So there's phosphate groups are negatively charged, choline groups are positively charged. Then you have a bunch of calciums that are helping link to the negative charges. You can break this up by adding salt, changing the pH, or adding a chelating agent to basically take away all the calcium, like EDTA. Now the lipids, there are a lot of complicated lipid anchors. You don't really have to know much about them. You just have to know that you can covalently attach a fatty acid or a phospholipid to a protein, and then you've got a way to anchor that into the membrane. And just look at this one right here. This is the GPI anchor. And the GPI anchor is actually a phospholipid, so it can be broken down by a phospholipase. There's a whole bunch of other ones. You don't have to know them. This is just given you as an example. And the GPI anchor is one that I've seen in a lot of places, so it's probably important to know that that one is an important exterior linkage, and there are many proteins that can be linked to a cell that way and can be specifically broken down by breaking that. When you look at it all together, there are different types of integral membrane proteins depending on how many chains they have. So if you look at these, I want to focus your attention to the four on the right, which follow a certain pattern. Type 1 actually have the NH3 group outside and the COO group inside, and they just have one single membrane-spanning alpha helix. Type 2 is exactly the same, but it's flipped. Type 2 has the COO on the outside. Type 3 has a bunch of different helices, but they're all in one chain. And type 5 has a bunch of different helices with those helices in different chains. So technically type 5 would be a type of quaternary structure. Type 3 would be a type of tertiary structure. Those are the only types that you really need to know. So just remember, type 1 starts with the amino terminus outside and has a single helix. Remember those things. Type 6 and type 4 are just weird to me. But the reason why it's important to be able to talk to someone and they say Bacteriorhodopsin is a type 3 integral membrane protein, and you know what that means. What it means is that there's a single chain with multiple helices, and so it wraps back and forth inside the helix. Bacteriorhodopsin is important because this is a version of the same protein in your eye that holds retinol that allows you to see. But it's really interesting. I just told you right there, the protein that allows you to see is a membrane protein. How does that work? Well, that's what later in the book Biochemistry 2 is going to talk about. But there's a hint that membrane proteins are important to letting you see. This actually lets you see by opening a door for protons to cross the membrane or other ions to cross the membrane. It's actually it's a channel that allows ions to cross through. So the reason why we knew really early on that rhodopsins are type 3 proteins is because you don't have to have the crystal structure to know that they are a type 3 protein. All you have to do is have a look at what's hydrophobic and which residues are hydrophobic and how many in a row you have. Here's a hydropathy plot that shows that if you take the residue number and you say how hydrophobic is the stretch of residues that's like neighboring that particular residue. So you look at the residue and its neighbors, you see how hydrophobic they are. And you see, if you do this, you see that you have stretches of about 25 to 30 amino acids that are really hydrophobic, and you have 7 of those stretches. Those correspond to the 7 helixes of Bacteriorhodopsin. You can find this out just by what you know about amino acids, and it's why I made you memorize so many amino acids early on. Now you can just look at a sequence and you can say, hey, that's a lot of amino acids. They're all hydrophobic. There's 25 in a row. That might be a membrane anchor for a protein helix. If you looked at a protein and you looked at this, here's glycophorin. So look at this one and tell me how you can interpret it. I'll wait. You should see one hydrophobic 25 residue stretch that looks like one alpha helix that goes together with the protein, that holds the protein in a membrane. You can even tell it is a type 1. Actually, you can't tell it's a type 1. I take that back. You can tell it's either type 1 or type 2. So if you look at membrane proteins, you can see this pattern. Hydrophobic residues will be inside the pattern, inside the protein. Actually, there's another little pattern you can see. If you color the charged residues blue, you can see that charged residues are outside the membrane. I want to be clear, this is outside the membrane, not outside or inside the cell here. But look at the water-lipid interface. They've colored tryptans and tyrosines, orange and red. There's a lot of tryptophan and tyrosines that are sort of at the interface between inside and outside. Inside, you have a lot of gray residues, which are uncharged. And so it looks like you even have a tendency to put the half-charged tyrosine and tryptophan residues at the interface, whereas things like phenylalanine, leucine, isoleucine will be completely submerged and in the inside of the membrane. So you can make proteins do different jobs. You can make proteins be transmembrane channels, like we said before. You can also make a big protein with a big hole in the middle to make a big door in the membrane. So you can make a beta barrel that stacks up like this and circles around, big enough to hold another protein in it. An entire protein can fit inside, and you can have these complex doors that let things in and out of the cell. Proteins are what do the jobs of letting things in and out. And so you can see a lot of proteins that follow this. They use beta strands, and it's called a beta barrel motif. If you look back and forth, you can see a lot of kind of cool looking structures here. But the one that I want to point your attention to is the one on the far right. It's called Hemolysin because that protein has a big hole in the middle. In fact, that hole is so huge it's going to be hard for it to close up. And the hemolysin is in fact made up by something that does not want to close up the membrane. It wants to open up the membrane. Take the name apart. Hemo means blood related. Lysin means to lyse. And so that what you have is you have a protein that is made, it's a toxin. If you look down at the bottom of the name, it's a toxin that will lyse red blood cells by punching holes in the membrane. You can see the hole right there. In fact, this is how it works. It's actually made as several subunits. The subunits hold this little orange beta strand, this beta hairpin in them until they get to the red blood cell membrane. Then they latch it out, they make, and they find other ones. The monomer makes a heptamer that comes together and makes a big pore that punches a hole in the membrane. This is how hemolysin works. And as you can imagine, it's bad news to have this. It is toxic to blood cells because it literally punches holes in them by using this kind of association mechanism. The other thing, now that we can see membrane proteins, we can find their structures, sometimes we don't see just proteins. Sometimes we see that there are membrane lipids that are stuck so tightly to the protein that they're actually crystallized in the crystal structure. And you can actually see where the membrane goes as a result. For example, sheep aquaporin. Look at this. It has all of these long fatty acid chains, and then it has all the head groups on the top and bottom. Head groups are blue, fatty acid chains are yellow. You can see how it fits into the membrane because you can see part of the membrane still stuck to it. On the other hand, here's a big pore membrane, which is called F0. It's part of the complex that makes ATP, and we'll talk about that in Biochem 2. If you look at this, you can see the membrane is still stuck to the protein. These are called annular lipids, when you have a lipid that is still stuck to the protein. And it tells you something. It tells you that the lipids are stuck really tightly to the protein where they come up right next to it. The protein makes the lipids a little more solid. And that means that lipids are going to be flowing more freely when they're away from the protein. Annular lipids are going to be frozen in place. So one of the things, the reasons why I did this, remember I showed you Cardiolipin, and I said that it was a weird kind of four-legged lipid that was connected, had two connected head groups. Well, the reason why it has two connected head groups is because it is an annular lipid that actually sticks to proteins and it holds protein complexes together. The other place we saw it was in the mitochondria. Mitochondria have all these machinery that sort of work together, proteins working together in the membrane to do all the stuff that mitochondria do, to make energy for the rest of the cell. Cardiolipin helps hold those together by literally stapling them in place, by binding one protein with one end and another protein with another. So I think that's just a really cool way to use biochemistry to hold proteins in place. So the main thing is that's what I want to leave you with. There are islands of different lipids. There are microdomains in the protein. And we have these different areas that flow differently. And remember that you have lipid rafts in this as well. Here's literal lipid rafts, and you can see this as a microscope image where it measures the thickness of the membrane. These lipid rafts not only are different membranes together, they're different thicknesses because they have different longer carbon chains to them. And the membrane is literally thicker at this point. So if you look at it from the side, you can see that these longer, these lipid membranes are going to be enriched in sphingolipids and cholesterol. Cholesterol likes to dissolve in these. And so you have this area, and this area itself floats around the membrane. So membranes are these amazing fluid structures. The other thing that I'll just point out, if you have a GPI anchor, the GPI anchor has a long chain to it. So the long chain will be associated with these lipid rafts, and the lipid rafts might move around in response to certain signals from outside the cell, which will change how the cell works. And how it all comes together, we're still figuring out. But we're seeing that there are these different areas, and it's based in the basic biochemistry of how long, how thick is the membrane in that area, how many carbons are in the fatty acid chain. One of the really cool things is they've seen how this is something looking at just how the hydrophobic effect works. Just from the hydrophobic effect, you can get these little whirlpools where all these long chains come together, and then they tilt because they don't quite fit with the rest of the membrane, and then they swirl around, and they literally make these little whirlpool vortexes in the membrane. And as you change the temperature of the membrane, you get different amounts of these vortices. So at 28 degrees Celsius, you can see right here, part C, you have this rainbow-colored moving vortex that comes from the lipids swirling around each other. This has got to be used by the cell somehow, but we don't know how yet. I just think it's really cool. And all of this comes from oil and water, the hydrophobic effect working with the way that membrane lipids come together. That is all I have for you. So that's the homework problems. This finishes up chapter 11. Make sure that you do all the stuff on sapling. It's all listed there, and HW11 is where it is. So go ahead to that, and be aware that with the long weekend we also have a reading quiz, but it is the last reading quiz of the year. Reading quiz for chapter 8 is due on Monday. We'll be talking about DNA then. But thanks for listening, and I hope you have a good Thanksgiving, and I will see you on Monday so we can be thankful for biochemistry a little more together, as the case may be. All right.